My name is Therese Coleman. I'm a senior. I am studying cell molecular biology, and I have a minor in biochemistry. So, molecular research is a very slow-moving process. So, we spent over a year, or well, I guess about a year, just inserting the C2B9 protein into yeast cells to get the yeast-2 hybrid screen to get the identities back of proteins that interact with it. But that doesn't really tell us much about what's going on in the cell, specifically with C2B9, which is what our research is about. So this is the next step to really start to understand how it plays a role specifically in mammalian cells. So because it takes so long, because we're working with like yeast cells and bacteria and they take time to grow, and sometimes they're very particular and we have to repeat steps if the yeast aren't happy and aren't growing as they should be. So being able to continue this to see the next step of what could actually happen in a longer term than just one summer is really beneficial because I get to see the process from the very beginning of when we started identifying kind of what we were interested in and how we wanted to go about working with C2B9, and now seeing, okay, we have these results, what can we do with them? And we work to identify that the next step is to insert them into mammalian cells to understand their interactions better and see if they even do interact in mammalian cells. Because with the yeast-2 hybrid screen, they might be interacting with one another just because the two proteins happen to be in the same yeast cell because of pure luck, but that doesn't mean they would actually interact within a human or any other sort of mammalian cell. So this gives us the perfect opportunity to continue it and see if our research from last summer is the progress that we want it to be.